Silvery, large eye, sort of blunt snout. And so that's my best guess as to genus. I have not seen this particular species before. This is a new one for me, so I'm just, again, guessing here.
Okay.
But they're beautiful deep water animals.
Let it go.
Oh, right. Thank you, Chris. Yeah, there are a lot of areas of black staining. I'm going to call it staining, and what it really is mineral deposition as a consequence of these warm fluids coming out, hitting seawater, and then
the Rida lasers. Looks
what happens is that
different,
the
fuzzy
metal sulfides
go for zoom.
Begin to precipitate out of the seawater. Looks like there's maybe a little finger right above where we're looking at. And look how close those hydroids, or whatever those little animals are that are growing out the seafloor that are hairy.
Spires just to your port. Right.
It's the original one, right?
Okay, great.
Yeah. Thank you, though.
I don't really know what the Ted is doing, but I'm sure Jim's on top of it.
Okay, watch lead, are you ready to move on to find new spires?
We are ready to move on. Thank you very much.
All right.
Can you fly across the top of this one?
I can fly up it for you, but it's not in the direction of motion here. If you want to do a partial zoom to get the skids out.
As we rise up this, you can see that there are individual fingers of this chimney structure that are poking up into the water column.
This stuff isn't too hot. We should be able to fly right over it.
Push the blacks a little bit.
This one's not, or this one doesn't have anything anyway.
Okay.
Lots of corals. We actually should image a coral sitting on top of this.
All right, I will leave that to our trusty new pilot, Dan. Doing a little watch change here. Thank you, watch lead.
You are welcome. What we see in the top of the screen are fingers of this chimney structure pointing upward in the water column. And that's because the fluids that come out, they do cause precipitation of crystals in the water, and then those harden around the vent itself, and the entire structure grows upward as venting continues to proceed during a really active phase of fluid coming out of the seafloor in structures like this. So it kind of builds up a huge deposit on the seafloor. And as I was saying earlier, in volcanic terrain, particularly around island arcs, if there are a lot of these vents on the seafloor, they get covered with the ash from the explosive eruptions, and that makes an insulator blanket on top of these. So over time, these deposits can enrich themselves. There's a particular type of deposit like this in Japan called a Kuroko
left-hand edge
deposits. These ore deposits are incredibly rich.
Welcome, co-pilot.
Thank you, navigator.
I'm sure you were informed, but just in case, there seems to be an error
shore side. Notice the crab
D2
off in the
depth sensor.
distance.
Yeah, 10 meters.
D2 is about 10 meters shallower than indicated.
Copy that. Keep an eye on the delta.
We saw
a delta of
crab
33
off in
earlier.
the distance.
That was a little strange, but
our videographer saw a crab, and that's not something I recognized.
Granted that is greater than the tether.
It's greater than the tether length. That is correct.
Let's see if anybody on the chat has noticed it. No. Tim Shank says it was near where the last fish was imaged.
Hi, Patty. It's Scott.
Yes, Scott.
Okay, I just wasn't sure if you could hear me.
Yep, I do.
I think I see the crab in the far back right, but it looks like you were just reading the comments from Tim Shank about this possible dandelion, and that would be interesting to see. I don't know that we've seen any on this leg yet. It would just be a very interesting contrast to the stylaster and hydrocorals because they're both hydrozoans.
Okay, we're just having a pilot change here. And if we aren't going to be moving real rapidly out of here, so as soon as they get settled, can you tell me if it's the yellow-looking object on the right of center of the screen, just slightly above the center?
Yeah.
Okay.
I see what you're referring to. I do not believe it is that yellow thing. It would be a more consolidated kind of orangey ball.
And I
did not see it. I was just reading what Tim wrote, and so I'm not sure where we are with respect to the fish.
Yeah, I'm not sure either. Well, as soon as the pilots get situated and we get a little closer to the seafloor, we can take a look around.
Yeah, I understood. That's the only reason I suggested it, because I heard there was a watch change, so I knew he would be sitting here for a little bit.
Yep, that's true. Our videographer probably can give us a clue as to where that was. So maybe I'll rely on him to give us an idea.
All right, watch leads. Just had a shift change. We're now going to have Andy Lister as navigator, and I'm Dan Rogers as pilot, and I have next to me Levi Unema as co-pilot, and of course Roland Bryan, video engineer on the camera, and Annie White back in the clipping station. And we're all with the Global Foundation for Ocean Exploration, and excited to hopefully show you some cool stuff this afternoon.
Oh, no doubt.
So what's the first thing you want to look at?
No doubt we will. Well, one of the things we were trying to find again was some biological samples that I'm hoping that maybe our videographer can guide us to, because it was near where a fish was imaged earlier, and I'm sure that fish has moved.
Okay.
So upper
Patty,
left,
are you asking for a biological sample or some imagery of bio?
They were indicating a dandelion. I did not see
We're
the
asking
dandelion,
for
but that's where we
an imagery.
Fish.
We have not yet
So
collected a biosample.
to your left.
Okay, going a little left. And so watch leads, you want to look at the spire again here?
Casey, we're
We
trying to
have
find
looked at the
something
spire.
that,
No, thank
Tim
you.
Shank noted in the event log, and none of the rest of us saw, and we'd just like to see if we
So
could see it
co-pilot ship
before
is
we
stopped.
We got
Everything's settled out. Just keep an eye on
Yep,
that.
totally understand. I just wanted to make sure that
Delta spire.
it was not
And
a
the delta's
request to
10
stop
meters
and
off.
sample something.
So, and that's an estimate. So.
But thank you, Scott.
Yeah,
Correct, understood.
my altitude helps me out a little bit. But yeah.
Yeah, watch lead, pilot.
Go ahead, watch lead.
Yeah, pilot. We've taken a lot of video of this chimney that we're looking at in front of us, and maybe what we'd really love to do is take a look at the biological object we are searching for. I guess it's to the left of the spire.
Okay. All right. Well, ship has stopped. Everything's settled out. So I'm going to move over.
Okay. Thank you.
And now, if you just want to kind of get your bearings and determine a general direction once we want to get moving, so we can be ready for that.
I'm going to have a quick
Things
conversation
they were
with Watch
describing
right
was
here.
a dandelion.
Okay.
I did not
Hey, watch
see
these
it,
now.
but they indicated it was near the fish that we had imaged.
Go ahead, Nav.
Hey, since I'm just changing it here, I just wanted to get an update and what your general plan for this is. Do we want to get over to waypoint 3 and waypoint 4, 5, and 6, or are we just good exploring and slowly making our way up there?
What we're doing right now is just trying to find this dandelion, and then, yes, we do want to go to the waypoints that you mentioned.
Okay.
And as soon as we have found this critter, we can get on and do that. Thank you.
Okay, well then I will keep us kind of moving after we have found this and in that general plan of getting waypoint to waypoint.
I figured we were zoomed in a little bit.
Yeah, that's great. Thank
So
you.
was
Copy
past
that.
the skids. You want to back past the skids?
Let's keep it wide while we. Oh, what is that?
That's
That
that lobster finally.
Is one of those
zoom in.
lobsters.
We've seen him a few times today.
We've
but
seen about
always
6 of these
in
guys
position.
now.
Cool. I've never seen that before. That's neat.
They
All right.
are really nice.
Zoom out. Keep looking for your. You believe it's a dandelion?
Yes. Best place I can direct you is near the spire.
And I believe it was to the left of it.
All right.
Tim Shank says it was at the base of the chimney.
Was it at the base of the active chimney or base of the one we are looking at right now?
I thought it was the active
Do you want to go
chimney, but
partial?
I'm not certain.
Hold there.
Okay,
So this is the inactive one.
this is the
You don't
inactive
need to
one.
spend too much time trying to find it. I mean, it's a small thing. So if we don't come across it as we pass by, we just move on.
Okay, so folks on shore said if we do not
Seeing
see
a
it as we
hydroid
pass
right on the
by,
edge of the screen,
we
bottom
can
corner.
just zoom on.
Okay. All right. Well, what's our general direction here? Is
Looks
it
like,
north?
yeah, it's about northwest.
Colorado's coming in.
Last one was 340. We're going to
Okay.
keep around that, between 320, 340.
Can we zoom out the video before you? Hey. Thanks. So it looks like 340.
Yeah, between 320 and 350 is going to be our general heading for the next while.
Okay. Well, let's. We want to, give ourselves plenty of room around the spires. So
Copy that.
if we can go a little more north of that, I think as long as we just end up on top of the ridge.
Yeah, we can definitely head in a little heck, we can even set in like 10 degrees.
Yeah. Let's do that. So let's see. Just keep a good eye on your tether, and I kind of want to stay under you so it's not blowing off to the side into potential other spires.
Copy that.
Got some returns on the sonar.
We're doing pretty good on time. We have to average 0.08
Yep.
over
Okay.
ground.
So inside the
That's-
crater over there.
to get to roughly waypoint 6 in the end.
Copy that. Navigator.
So for
But we
scientists on shore,
probably
if you look at the quad
you
screen on
have,
video feed 3,
50
there's a
degrees
high pack screen.
pilot.
If you can see
Okay.
waypoint 2,
Just gonna
that is the
look
edge
out
of
a
the
little
crater, and waypoint
Copy
3
that.
is where we will
Yeah, let's
land
be
once
very
we drop
mindful
off
of
the
where
crater,
the tether is.
just for
Yep.
your awareness.
And that will be inside the crater at the top of this seamount. Certainly
All right,
looks
so we
like
got
there's
that
a lot
feature on
of
the
hydrothermal
left.
action along the
But it
crest.
looks like it's not, there's no major features 30 meters out to my starboard here.
We got your sonar at 30-meter range right now.
Yep. So let's do, let's go ahead and do 20 meters, and why don't you turn your head over, co-pilot?
Closer to north.
Yeah.
Coming northbound here.
We may want to be a little safer and even go 10 degrees. I mean, it's up to you, pilot, though.
Yeah, I'm looking at like I'm looking at north right now.
Yeah.
So yeah, why don't we, actually that looks alright. Let's do 30 meters due north
Okay.
for now.
Yeah, I'm happy with that.
Just to give ourselves plenty of room around these.
Copy
All
that.
of these little black
Okay, coming down.
spots
Very
on
Darby
the video screen are very likely individual
Hey,
places
Bridge, we're gonna
where
go ahead
warm
and put
water
your
is
ship
coming
here
out of the
30
seafloor.
meters at 000 degrees. Decimal 2 knots, please.
Got all your lights stowed right now.
Great, yeah.
You want to keep them in right now?
Let's keep them in for now.
Okay. I'll turn
Good copy.
hydraulics off then.
Thank you, co-pilot.
Yeah.
I'll just kind of turn and look here. I guess while we're waiting to get moving, I can...
There's an interesting sort of curving lineament along which a lot of these black patches are forming on the right-hand side of the screen. So that could be
What was the height of that
local
spire? I don't have it in here.
deep penetrating crack
It's taller
in
than
the
D2.
material on the edge of the,
Wow.
Not by much, but-
crest of this caldera rim or crater rim.
Yes, maybe.
Oh,
maybe
you
10
can see a round log-like looking thing on the right-hand side of the screen, and that would be one of the tall fingers of this chimney that have broken off and fallen onto the seafloor. And probably the log-like feature that is going from upper right
Looks
to
like we're
lower
starting to move,
left,
pilot.
kind
I
of in the
like to stay
left-hand
20 meters off the
corner
ground just so
of
the
the
tether
bottom
is a
part of
little
your
bit
screen.
higher.
That would have been
That's
the
fine.
top
Okay.
of the thing because it's a
And
bit
pilot,
narrower.
It was noted to me
So it just
that
fell right
our
off. This thing must have been
depths
pretty
do
big
not
when it
match
was
up.
Yeah,
active.
the delta Z is about 10 meters off right now.
Yeah,
Over on this one.
D2's depth meter.
So our altitude is fine, but our depth sensor is off, and our delta Z is off by 10 meters.
In the positive or negative?
We have 10 meters more. It's actually 20 meters, even though it's reading 30.
Okay.
Might need to adjust my Z-bias here a bit. Okay. Do you want to do a partial zoom, Tara, on this? Now that we're kind of set up here.
Absolutely.
Okay.
Going tighter?
Yep. Yep.
These aren't.
Okay, we're seeing something.
These aren't right.
Back from lunch, there is
What's not
an
right?
encrusting sponge
Well,
at the
like
top
my contour lines match
and
up.
hydroids.
I think
And-
when
No
we
continuity.
What do you mean by that?
Well, because we seem to be missing one here. I need to just follow it around to see which one we're actually at.
Okay, let's zoom out.
Coming light.
it's 890, right? Yeah.
Okay, just styling my Z-bias here, watch lead.
That's right.
Okay.
Okay.
That one's
Just noticed I was floating a little bit.
Okay, no problem.
All right, okay. So the ship is moving. We're moving north up the ridge. Is there anything in particular we're looking for?
Well, more of these chimneys for sure.
Okay.
I don't see any.
I don't see any in the
There's nothing
sonar
in it.
right
Well,
now.
There's maybe a depression over there on the right.
Yeah, I see that.
Nothing. No big ones in serious.
Yeah, nothing
SD2
view yet.
Yeah.
Look up here. Yeah.
Yeah, there's that. Yeah, I had a depression there.
Okay, lift up, push out.
So we're heading into the crater now.
We are.
In
I
on
think
the
still
ridge.
we're still
on the
heading
rim.
up
Still,
the side of the outside
okay.
of the ridge. Yeah.
Still on the outside of the ridge.
It looks like we might be getting to the top.
Yeah. They're at 894 meters.
The ship is. Looks like that move's probably about done. Let's
I'm still swinging, but
We got
yeah.
another 5 meters.
Yeah.
Yeah, I'm at just due north, position of direction of movement, pilot. You're on my right side.
Yep. Kind of looks like it slopes up
Just a
to the
little
left
bit.
here. So I'll come back into your view.
Yeah, navigator. What's our bearing to waypoint 3?
Waypoint 3.
We're going to be
right
in the
now
middle of the crater.
Yeah, right now we're still trying to get to waypoint 2. But after that,
I think the idea is we're just going to get up to the top of this ridge, and then we're going to look at the slope. If it's subtle enough, we can try to creep down the back side of the slope, but if it's steep with potential spires, we're going to just hop
Yep.
to waypoint 3 in the middle.
well, we're looking about a bearing
Yep,
of
roger that.
310 then.
Excellent.
Okay, good.
Thank you, navigator.
Okay, let's keep the ship moving. Let's see.
That was a 30-meter move last time due north.
Yep.
But your sonar, I mean, it looks like you're getting to the top facing where you
Yeah,
are.
let's do another, do another 20 meters.
Okay.
So for those of you who are just joining us, we are on the NOAA ship Okeanos Explorer, America's ship for ocean exploration, and we are coming live from a place called Chamorro Seamount. And we're exploring unknown areas of the Marianas Trench National Marine Monument. This expedition, it's called the 2016 Deepwater Exploration of the Marianas, is possible through successful partnerships and collaborations. This mission is designed and supported by NOAA's Office of Ocean Exploration and Research, and the ship is operated by NOAA's Office of Marine and Aviation Operations.
20 meters, that's 20
it's part of a
meters
3-year campaign to address the Pacific Monument's science, technology, and ocean needs. So it's called Capstone. And the Capstone mission was designed to address the fundamental need of resource managers to better understand these deep parts of the nation's marine monuments.
I guess we might notice a bit more current once we get up top here, so just keep an eye out.
There are a number of partners, key partners in this effort.
We still have a,
within NOAA, in addition to NOAA's Office of Office of Ocean Exploration and Research, which is within the Office of Oceans and Atmospheric Research.
You see something on your sonar there.
We have the National Marine Fisheries Service, NOAA's Marine National Monuments Program, and NOAA's Deep Sea Coral Research and Technology Program. In addition to that, a very important partnership is with NOAA's National Center for Environmental Information. And through these partnerships, we're able to collect all these data, process the data, make the data publicly available, actually in a very short period of time, within 60 to 90 days after they're collected. My name is Shirley Pomponi. I'm one of the science leads on this mission. I'm from Harbor Branch Oceanographic Institute, Florida Atlantic University, and Patty Fryer just ducked away for about 10 minutes to go get some lunch. We made her tear away from the screen so she could do that. She's from the University of Hawaii.
Go ahead, Mike.
Hey, video. I was just wondering if
running
these
the ROV operations
over here are still hooked up over there.
Are crew members from the Global Foundation for Ocean Exploration.
Is there anything we need to do to get him hearing it?
As soon as they get a minute, we'll have them introduce themselves as well.
Yeah,
they just did.
We just did, but
okay, they just did. Never mind. I just asked if there had been any intros. I was out having lunch too.
That's fine. Thanks for.
Hearing anything?
Okay.
Thanks for remembering that.
Okay.
Tell me if you hear anything. Nothing.
Okay, so we're seeing
he's not hearing anything yet.
Some stylaster corals.
Copy that.
That looks like it's the dominant fauna here, at least the dominant macrofauna that we can see as we're moving across this.
Okay, so I think what we're looking at our starboard is that slope up the crater to the right.
Yeah.
But it looks like this is kind of the top of. Ooh, there's a fish there.
Look at fish over there. Yeah. Yep.
This is definitely the local eye.
Yeah. So this is probably the top of the ridge.
So
here.
for people that are just joining us,
cool.
could we get a brief overview of sort of where we are and what we're doing and where we are in the dive, what we've seen so far?
Yes, we are at a place called Chamorro Seamount, and what we were. This is an area where I guess there hasn't been any volcanic activity reported recently, but we were really interested in seeing what was going on at this area. Wait, before we go into that, let me see. Is there Bruce? Are you on the phone? Yeah, Bruce. Why don't you tell us about this? It's a rat tail.
Well, that's about
That's about it, right?
It's a rat tail. It's a different species than we've seen before. Chris had typed in that it could be in the genus Coryphaenoides, and I agree. It's not a species that we've observed previously, though, because it's got the white tip on the dorsal fin, the tall fin sticking up on the back. You can see it's got kind of a concave head, a pointed snout, very tall dorsal fin, fairly large scales, and a distinct twister on the chin. So, again, as with so many of these fishes here, we'll have to do some work, look at the images and compare them to descriptions of different species to really figure out what this is. But it's obvious from this dive that these rat tails are a dominant type of fish at these depths. I think we've seen 4 or 5 different species of rat tails so far on the dive, and this one is different from what we've seen before. And it's also different from what we had observed in the Hawaiian Islands. So we have another puzzle to figure out. And
okay,
that's
so
the
I think we're on the top
exploration
of this ridge in this direction.
Okay.
And again, the next one is about 310.
When we get the images and
toward waypoint 3.
Towards waypoint 3.
Okay, let me, once we're done here, I'll lift up, look off in that direction, and we'll determine how steep the slope is.
Thank you.
That.
Thanks, Bruce.
All right, let's zoom out video. Great.
So as this mission was being planned, we had meetings with the
over 180.
Scientific community to identify
so that might be
sites that would be of interest to explore. This particular one was proposed, and we decided we as the scientific community decided to do some exploration here. It has no known historical eruptions, and there was one dredge sample that was made back in the 1980s, and there were igneous rocks that were collected. So the plan is to explore this seamount, including where we are right now, the summit of it, to see if there's any volcanic stratigraphy, and sample the rocks to see how it's compositionally evolved, and also to explore the biological communities at the greater water depths.
50 good.
50 is fine.
So we saw some really interesting biology when we first got into this dive. Some things that Chris Kelly at University of Hawaii was telling us that we very rarely see. We saw 3 blind lobsters.
Let's go ahead and head to waypoint 3. So
many other really interesting sponges as well.
We'll do a 30-meter hop. I'm gonna just watch the tether, watch my tail end.
5 science themes of this capstone
Let me know if I'm getting out too far ahead, because we just want to make sure the tether isn't going to drift into any chimneys.
Characterize seamounts that may contain deep-sea minerals.
You guys ready for that?
To investigate the geologic history of the Pacific seamounts by collecting rock samples, identify and map vulnerable marine habitats. So we're doing this by getting high-resolution video imagery of biological communities, and we're collecting some representative samples when we can. We're acquiring data to support priority monument and sanctuary science and management needs,
a little bit of a current here,
and increasing our understanding of deep-sea biogeographic patterns across the central and western Pacific.
It may whip around in that channel.
We
when you get down into
are certainly collecting a lot of baseline information, and now in all of these deep water areas.
Another fish out there.
Today we're specifically focusing on exploring some new potential hydrothermal vent sites and seamounts.
Do you want to do a partial zoom video?
Sure.
Hold there.
Okay, so again, some more of these stylastrid corals. And looked like was there a crab on the back side of that?
Squat lobster.
Squat lobster on the back side of that. And some of the finer branching things that we're seeing over just underneath of the bright white corals are hydroids. And coming around, we can start seeing those pincers.
Can we kill the lasers?
Of the squat lobster. Yep,
there's 2 squat lobsters, one up at the top and one down below that just ran out of the video frame.
Oh yeah, I can see that. The one kind of in the back also.
He's a little bit larger.
Mm-hmm. Tim Shank from Woods Hole is saying this is the stylastrid seamount. Indeed, that is probably the most abundant, certainly the one that's the most obvious fauna in this whole area here.
Cool.
Very, very abundant. Of course, when you look closely, you can see all those hydroids as well.
Okay, zoom out video. Thank you.
Nice pirouette.
Thank you.
Oh, yeah. So
this
all
is
those,
cool.
Yeah.
Do you want to brighten up the background, Tara?
Yeah. So all of these those white blotches that you're seeing in your screen are all stylastrid corals.
No, we'll keep it moving.
Because
we're not even halfway through our move yet.
all
That's fine,
the
we'll keep
pinnacles.
moving.
Yeah. Oh,
But
yeah,
these
there's
are
more
all-
pinnacles back there. Yep,
I
look
think
at
you
all
got a
those
spire back
pinnacles.
there in the back.
Yeah, there's a big one
Yeah,
there.
there's a bigger one back there.
These are all tether hazards. That's what we're gonna think about here. So
add just something
keep very
that's so
mindful.
interesting about the range of
staying way
possible
above you to
morphological
thanks.
diversity
Yep.
in some of these groups. So we're talking about these
These are quite large, huh?
stylastrid
Good ones back there.
hydrocorals, and you also noted
Stronger
those very small hydroids. Those are both in the same class. They're
Exactly.
both hydrozoans.
Yeah,
And
right.
that would be why we really see a dandelion animal,
Yeah,
because
pilot, I'm
those are
starting to
also
move, and I got you at
hydrozoans,
6
and so are
little note
the
here.
siphonophores and so on. And basically, those stylasterins are very hard with a hard calcium carbonate skeleton, whereas those other hydroids, the very small ones, are very flexible with a protein skeleton.
I'm just going to push out here a bit.
Ultimately, they both
keep an
have
eye and
polyps,
let
they
me know
both have
if anything
tentacles, they're colonial, and so on. So
makes
they
it
share a lot of
uncomfortable.
similarities,
Yeah.
but they can also have a range
I'm happy over here.
of great
I
differences.
got a good altitude.
Yeah, that's. Thanks for pointing that
Looking
out,
down
Scott.
at
And the
you
other
at
thing
about
too is that you mentioned
80 right
earlier
now,
that they're
so
also
we're not going
related to
fast.
Portuguese man o' war. And so now
Yeah,
when
that's correct.
right, and think about these like fire coral, the stylasters, the hydroids. Those are all really nasty, stingy things.
So, Scott, is there some adaptation about hydrozoans that would allow for them to
Okay, it
live
wasn't quite as
in
tall as it looked
either
originally.
colonize these things quickly or live in this sort of
All right,
extreme
my turn.
condition? And I use, it's not necessarily super extreme,
Where we're
but
at here.
different than normal.
Yeah, Casey, not
Oh,
that
what
I
is
am
that?
familiar with. I
Zoom
do
in,
believe
zoom in.
you start to see more stylasters as you get
it's
to
south or
shallower deep sea depths. So,
Siphonophore.
we're at the
Siphonophore.
Really
100
interesting
meter,
fish
800 meter range
Oh.
today rather than,
Crazy.
deeper than 1,000 meters. So that
can get
could
this
be
out if we
one
want.
reason.
Yeah.
It could be
So
something about the
Got about
temperature.
10 meters left.
I'm really not certain.
Thank you,
I'm not
Nala.
an expert by any means in the hydrocorals, so I'm not sure what it
Floating
is
that
that
way.
controls their distribution. I'll let you go to talk with this.
So, as you were saying before,
actually,
So those are all
Fantastic. This
single
is a
cells
siphonophore.
making up a community? Is that what?
Right.
So
So
this is a pelagic version of a hydrozoan that we've just been talking about, right on cue. And at the right side, what you're seeing are a bunch of individual zooids or polyps that are
It's getting closer
specially
to you.
designed to be
I have a visual.
float
Just don't want to
floating
back up.
structures, so they're filled
Yeah.
with gas.
made
Some of
lateral
them
right out
may
of the
be
way.
bell-shaped, like a jellyfish, so they can squeeze
Just
and
here to
therefore
make it
pump
some
jets
prop
of
wash,
water
push
out
them
for
away.
locomotion.
Yeah.
And then the stuff that's trailing out to
take
the
a bit
left,
more
and you can
zoom.
see
Go
the
ahead.
orange color in
Upsy.
there, those would be the feeding
cool.
polyps,
Hold
the feeding
there.
zooids. And so once again, this is a colonial animal, arose from a single egg or a single larva, but it continues to bud off little units,
Yeah, he's moving
and each of
faster.
those units in this particular case has a function. So some of them are for
Sorry, Tara.
buoyancy.
No worries.
There, that's a really nice view.
That's
You can
great.
see them pumping.
just gonna
Fantastic.
Hold there.
So those are each pumping out little jets of water, and so
Tilting up.
allowing locomotion for the entire colony. And then those ones further to the back will have the tentacles
gonna
and
zoom
the
if it's
stinging
all right.
cells
Go ahead.
that they
How we
can
looking,
use
co-pilot?
to
Looking back
capture
at you.
Okay, I need to push
their
ahead
prey
here.
items to
Okay.
feed. And then some of them
Let me
will
just
also
hold
have
this for
reproductive
a
structures. Just
seconds if we have
fantastic, wonderful imagery, and right on time.
yeah, and Scott, do you think those little balls there are bioluminescent?
And that move is done, or yeah, it's done, huh?
I got a little bit of
anything's
swing left in
That
possible.
it.
move
Okay.
is done, but
All right, I'm gonna push out. Let's do that video.
And,
Understood.
and Scott, these are more closely related then to the
Thank you. Yeah, we
got a
stylasters than they
meter.
are to jellyfish.
Absolutely.
Go
Perfect
figure.
observation.
Yeah, go figure. So again, really, really good example of the morphological diversity, even within one taxonomic group.
Right, exactly. It's just amazing what you can do with a few genes. I was just going to say,
Once
somebody
you
I
get
don't
out
remember
ahead there,
if
we
you
can
or
do
Casey was asking
same
about
move, same direction if
possible
you're comfortable.
bioluminescence. Some
Yeah.
of these siphonophores
Not much of a
are
slope. It's
known to
very
have
jagged
lures.
terrain,
The
though.
tips of the
Yeah,
tentacles
not
are
expected at all.
bioluminescent, and they sort of dance them around, and so
Never know
it's
what you're gonna get
possible a
down
fish
here.
or something
Yep.
thinks it's a little crustacean and goes
Exploration.
after it.
I like this shot. It's like D2's right in the center, you know?
Yeah, pilot, let's go look at that.
Okay. We can certainly do that.
I forgot to say please.
Oh, well, it's implied. Don't worry.
I got you at 75.
Okay.
Got plenty of time.
yeah, we're no longer moving, so
Yep,
you're
yep.
good.
See, now I'm seeing kind of shimmery stuff in there in the water now too.
We're never going to make it to the top of the crater.
Oh yeah, I see it too.
Yep.
Oh, it's coming out of there.
So
oh
what
boy.
is that
okay.
white
So
thing
let's
at the
make sure the
bottom?
tether doesn't get above that. Co-pilot.
Copy that.
Do you see it? It looks like something growing out
activity.
of that
Yeah.
little
I'm going to
chimney.
mark that here too.
Yep.
Exactly.
We're going to take a temp reading.
Yeah, yeah. Wow. Oh, wow.
Wow.
Wow, I know. Everybody's just saying wow. As we're
See
getting
how
closer and
CTD's doing
closer. Yeah, for sure.
Let's
I don't know if you
move.
can see this. See that
4.6 degrees.
Trying to think, 'cause
coming out of
I'm coming
that
down a
white
bit,
tip
so I might need
of this
Sirius to come a
cone.
little closer to me.
Oh,
So he's not pulling
some
my
sort of
tether as I set up.
calcium deposit?
So I'm still 4 meters up.
No idea.
Let's move the ship.
No idea. What?
About 10 meters.
270, something like that.
So I don't know if you heard the
5 meters?
pilot's
Yeah, let's
pilot
do-
earlier saying that this is
behind you.
they have to be really careful here
We're
about
trading
not
to
getting
5 meters
the tether
toward me.
wrapped around any of these
5 meters at
pinnacles. So these are all
305
kind of
degrees.
Yeah.
hazards working down
That'll
here.
give
You
you just
definitely
enough
don't want to
slack
get
to,
the tether
let me
wrapped
set
around
down
any
here.
of these.
Bridge, RV nav.
Yeah. I'd be
So these
comfortable with
also
that.
look a little different
And if
than
we need to move again, we'll
the
move
other
again.
spires.
They
These
do.
ones look
Just
like
being
little
cautious.
They look more like
triangles.
cones. They
we
look
need a
more
ship
like
move
a
of
cone.
0,
Let's go with the
5
zoom in
That are-
partial.
yeah.
0.05 degrees. 0.2 knots.
Hold there.
Oh, wow.
Look at that.
Okay.
That's pretty cool.
Wow. All right, so that's
No,
definitely.
I mean 0.5. Good copy.
So watch
Mary
these.
Jo.
We're starting to descend on the backside of the ridge.
Okay.
It's very jagged terrain, so
Apparently
we just want
there's
to
a
keep the tether
cuscule?
clear and safe.
Roger
So we're
that.
making a very small move toward that direction, and Sirius is going to come down and give me a little more slack to set down, and we can take a
Astrid, if
temperature.
you're on the telephone,
That
if you
looks
want to tell us
Roger that.
where you're looking.
I'm going to move on the opposite side. Actually, it looks like there's stuff.
Yeah,
Look
there's
like
stuff. What are those? What are those balls? Those spheres?
They look like snails.
We've already increased to 4.8.
Are they?
on
Yeah,
the
they look like
CTD alone.
snails. I do.
Why
They
don't you
look.
pull up the temperature readings
Oh
on your
wow! There's a
view?
crab.
Where-
There's a
what's that
crab.
Crabs.
There's
Yes.
a crab. I
There
think
you
we're
go.
starting
You
to
should- you
see
might have to
some
click two of
some
them if
vent
they're
fauna
behaving funny.
now.
They're
Tim
both
says
on
they're
right
snails.
now.
Yeah,
Double check
yeah.
It looks like
Maybe a
there's a
snail.
spike on
Lots
one
of
of them that's changing the
snails.
scale.
Albino chonka?
Yeah.
Yeah.
It's only at 12, so.
Albino chonka.
Let's zoom out.
Hairy snails.
Pull away.
Is albino chonka? Is that the name of this of the snail?
Okay, why don't you come down a few meters?
Well, usually
Coming
the
down.
prefix
Tether looks
albino is
happy.
from Alvin.
Okay.
Yes, I know that.
Yep. See all
I
the
just
tether?
wondered what the
That's what we
chonka
want.
part was. Wow.
Bring my altitude to
So
like 20
it
or
looks
something.
like these are
Yeah, that's fine.
typical
Okay.
typical
And
vent
I'm going to
fauna.
move to this side of it, away from the hot,
Yeah.
hot stream there.
Copy that. All right, holding at 20, 21. Somewhere around there.
Is that someone joining who'd like to
Yes.
give us a little bit of a rundown of what we're looking at right now?
Oh, concha. Albino concha.
Don't have anything there yet.
Okay, lasers on. So what is this? Well, is the white deposit at the top, is that going to be real fragile, or where do you want me to take the temperature reading from?
I would say take the temperature reading if you can, right up at that top, just over to the left of the.
Where we
That,
see the most shimmer?
yes. Oh.
Okay. Okay.
Tether looks good.
Yep. Sure
So
does.
Scott France, who I thought was going for dinner, he better grab his plate and bring it next to the computer. So this is albino concha.
I'll leave my temperature probe up. You have to your own
Let's see,
hydraulics.
albino concha hessleri, or hessleri, and it's of course named for the deep submergence vehicle
Auto XY
Alvin.
was not holding. Copy that.
And the species is named for
So I don't
Bob
want to touch down on
Hessler.
this.
The snails, these snails are typical vent snails from the Lao Basin and seamounts near Fiji.
Temperature probes are staying nice and chilly.
Okay.
5 Celsius.
CTD is at
So
4.7.
Astrid is pointing out that there's eels, but I don't see them.
I don't see them. She said they're over to the right.
Maybe in the background?
So we're betting that the temperature on this one's going to be a lot hotter than the last one. Look what you just got back here in time for, Patty.
Oh, I'm so excited. I took a lunch break.
A very short one. Very short lunch break.
You guys find good stuff.
So they're going in now to take a temperature.
Yeah.
Take a temperature reading of this.
Boy, there's a
and
lot.
You see the snails? The Alvinaconca hessleri.
I don't actually see it. I see
See
a
all those
crab.
balls? No, see all the balls?
Holy
see all those
smoke.
spheres?
There's millions of them.
Yeah.
And there's a crab in there too, right?
There's a couple of
Yes.
them.
Nestled in there.
Do we have an ID for folks on the line? Do we have an ID on the crabs?
What is the crab?
Scott said that the snails were first described from the Marianas Back Arc Basin vents. So.
And that is Mariana.
Mariana, sorry.
Back arc.
Mariana.
And it is the Mariana Trench.
Not the Marianas Trench, because you wouldn't say the Aleutians.
Or the Puerto Ricans.
The Puerto Ricans Trench.
Right, or the Peru's Trench.
But I mess that up all the time. The whole time we were working the Puerto Rico Trench, I kept saying Puerto Rican Trench.
Yeah,
It's a
but
common mistake.
a lot of people do that.
In defense of the common people.
Oh, sure. And in fact, to use the term the Marianas is correct, because that includes all of the islands and the region.
So I have to
the
say
Marianas Regional Trench National Marine Monument is correct. But you know what? I'll tell you a story later, privately.
Okay. So, all right, so I have a question. Does
Yes.
anybody know who the Queen of the Marianas is?
Oh, no.
That's you.
Woo!
Yes, we have a friend from Jams Tech. No, we have a friend from Jams Tech with whom you've sailed on many other missions.
Gee, that's true.
And he shared with us last night that you are known as the Queen of the Marianas.
Uh-oh. Uh-oh. That's as embarrassing as having the Friar Seamount named after me. Oh. Where's the happy birthday hat? I need it. I need it back.
Friar Seamount, we're going there next.
Oh, I know.
Not next. Soon.
So co-pilot, we're going to keep a close eye on the tether. Yeah. If you can go ahead and give me hydraulics, nav. If you can, if I need you to reposition.
Okay, we're going to get into position to take a
temperature. Each of these arrows is going to move you 10 centimeters in that direction.
That's terrific.
Yes. One click will be 10 centimeters in that direction. So I'm going to get the arm out and see if I can reach. If I can't, I'm going to back off. We're going to move, click it ahead just a hair, and we'll move, and we'll try again.
So all those little balls
please just
down
make sure
on
if you
the
can
lower
watch,
left
make sure that
snails?
the vehicle's not touching the bottom.
Wow.
So Tim
There's
Shank
no
is
hot
telling us
water
that there are
coming up to
actually
the vehicle.
2 species here.
Hydraulics up.
Ifremerie
Keep an eye on temp probes, both below 5 Celsius.
named obviously after
Nautilii.
Ifremerie.
Tether's happy.
Why don't you hand me the binder there?
Well-known
Predator?
Yep.
Oceanographic Institute in France.
And the white crab?
That's the darker, smoother shell.
Oh, okay. Okay.
The white crab is likely in
hydraulics are
the
up.
genus
Yes.
Austinagrea.
1500 RPM.
All right. Text out.
You can still see some
Speed back
stylasters
off.
over to the right. And
The white,
adjacent
the
there.
white guys.
Uh-huh.
Okay.
Yep.
Let's bring up Mini Zeus.
Patty, we
And
were
if you can
commenting
take that
before.
and
Oh,
follow
wow.
over to the
Oh.
Looking right down the throat of the vent in that camera.
Yeah. Yeah.
And actually, you can hold it there. Right there's fine. And actually bump it down and then bring the port wing inboard.
Okay, hold on.
Tara, is it possible to put that view on the screen so that the folks back on shore can see that? Okay, thanks.
Port wing inboard.
Yep.
How
Beat me
far are
to
you
it.
on it?
Let's see. Yeah, that's fine right there. That's good.
Right there.
Oh,
Yep, just so I can
it's
see
going to
it.
be
Zeus.
pretty
Wanna
easy
keep an
to
eye on
take
your
the temperature
temperature
measurement
probes.
Yep.
here.
Now we have two temperature
opening
probes,
is
one
kind
on
of
the
large,
vehicle
but
and
it's
one
a
on
lot
the end of
larger
there.
than the first one that we looked at.
So
There's
look over
one
at that mound over
on
on
the
the upper
drawer,
left.
the other
It
is
looks like
behind
a
Zeus
pile
Plus.
of those
Okay.
gastropods.
They did
there.
they drew it out a little
clustered
bit.
around.
Okay.
We were
Carl just told
commenting
me.
earlier,
just
Patty,
just be
that
careful
these
we don't
look
bump
different
it
from
into the
the
ground
pinnacles
here.
that we
We don't
saw
have any
before. These are
samples,
more
but
like
I'll retract the
mounds.
pin.
Yeah.
Always a good idea.
Looks like we're
Drifting a bit. Yep.
How's that for you?
So looks like there is
Gave you a
quite a
bump.
lot of hydrothermal activity up here on
Let's go
the
ahead more.
crest
Okay.
of the
Are you reading anything on the temp gauge
crater
already?
that we're hoping to explore. So we
it's
don't know
low.
what we're going to
Okay.
see when we get down into the middle
Hold
of it.
there.
Hold
It's
it
fine.
there.
Yep.
So the way these snails are distributed
Unfortunately,
will tell us
Respect
in
zero.
general
Okay.
where the fluids are coming out. So Albinaconca prefers the hotter fluids, whereas Ephremeri prefers the not as hot fluids. They're still hot.
Yeah, and they often compete for space, pushing one another aside.
Can we move the mini Zeus down, please?
Come down. I can bring wing in a little bit.
So the pilot's going to grab that little rubber ball
Oh, Zeus got
with
a
the
nice
arms
view.
of the manipulator.
Yeah.
And then the temperature
Nice.
probe is at the tip of the silver shaft that you see pointing up to
Are
the
you guys
upper
noticing the flickering
left.
on mini Zeus again? There? See that?
It's like a stripe go by to the center of the
Yep.
screen.
I've seen that 3 times in the last few minutes.
Okay, is that monitor
so the
or
That
manipulator
is,
is
seems like it could be a fiber issue.
giving a little bit of difficulty to the
We'll
pilots
definitely
here.
keep an eye on that.
And,
Okay.
you know,
Very
folks, that becomes an issue when
Excellent.
the
I'll come up with
pilots
you.
are trying to pick up a sample, particularly a delicate
You can take
biological
my Zeus
sample.
Plus as well.
Aye, aye. Got your Zeus coming up.
Tilt up.
So
we're not seeing barnacles.
We are
And
drifting ever so slightly.
Yep.
we're
Should
not
oscillate
seeing
a little bit, but
mussels.
should hold.
And we're not seeing the shrimp. So these are some of the typical
Okay.
fauna that one might expect to
Both
see
temp
here.
probes holding pretty low
So
right now.
everybody
They
be on
are
the lookout
updating.
for those.
If I can zoom my...
as soon as the probe gets close to this,
How's that temperature
you
reading?
folks on shore
trying
are
to
going to be
read it.
able to see the
So I'm definitely
temperature.
touching it.
You should be able to read a value.
24. Oh, yeah. 24, 26.
I see a value of
0.3,
26.39 degrees Celsius.
30.5.
Now
30.5,
31, 29, 28.
Get it in there.
Yeah, I'm definitely in there.
So you're
Okay.
right,
Those ships
Scott.
are all over you.
A lot hotter than the last one.
What's that?
Oh, absolutely.
What's our temp
Let's
reading at?
get it up to 30,
28.
27, 26. Going
27.
down.
It is titanium, so it will take a minute to stabilize.
Sure.
Could you bump me to the right, please, Nev? One bump.
One bump to the right.
Flickering on mini Zeus again. It's not a good sign.
We have a 37. 28
I'm seeing
degrees
the
C,
horizontal
29,
lines, the horizontal black lines.
29 degrees, 28 degrees, 26, 25. So the
Okay.
temperature is
Do we want to
fluctuating
measure
a bit.
anywhere else, or
So that's a huge temperature differential.
Yeah, there's a
It
huge
is.
temperature. Do we want to measure anywhere else?
Would you like me to bump
Nope.
you again?
No, I think we're good.
Yes,
Thank
please.
you, pilot.
Okay.
I think
Why
our
don't you
highest
back me
temperature
out?
was
Backing.
36.
Yep.
Back
36
one
point
bump, right
something.
one bump.
What was it up here? Like 48?
It's
Was
a
it
tilt down
48?
on Zeus. Keep an eye
I
on
think
that
the
now,
highest that I saw was 36,
Copy
but
that.
But this looks
That
like a big
looks
spike
higher.
up
Yeah,
here. It
you're
looks
right.
like 48.
okay.
Coming down on mini Zeus for you.
Thanks.
I can bring wing in too, whatever you need.
I'm going to bump one back.
So I presume that you have zoomed in on those snails already.
Actually, we have not, because we were starting to zoom in more on the shimmery
I think
stuff.
the front of the probe actually was touching the wing a little bit there. Oh, that looks good.
Did I bend the probe?
You can bend it back. It's fine.
Okay.
Yeah. We might need Sean to bend it back, but yeah.
Make it work.
Yeah.
Hold on.
Ooh, yeah. That looks like the right angle.
Tim Shank is telling us that from work that they've done in the region,
Happy?
that we would expect to
nudge
see
it?
snails, white shrimp, mussels, and polychaetes. Do we want to get a close-up of the snails?
Yeah, Pilot, we'd really like to take a close view on the snails.
I think you got right.
So
right
it's interesting not to see these fauna. What I see on my screen for the temperature looks like it's spiked up to almost 50, 40, certainly 42 or 6.
Yeah.
648. Yeah, like 49,
like 8
almost
or 49.
Yeah,
Right.
49.
I agree.
On it? Good.
Because it might straighten out when we bring the wing back out. Let's bring the wing out.
Wing, port wing outboard.
Tim is also noting that this is a shallower depth record for Ephemeri nautilii. He doesn't know if there are records shallower than 1,700 meters, and we are at 892 meters.
Yep. See both.
Okay. Pilot, when you get a chance, we'd like to zoom in and see the snails, please.
Okay. We'll get the arm stowed and we'll take a look.
Thank you, Pilot.
And let's bring the mini Zeus over.
We saw these same kind of snails way down south on the back arc
Want to
basin.
square your mini Zeus up when you're done?
Mid-Ocean Ridge Spreading Center area, and they were clustered around.
I know we have a few
vent
spares of those titanium probes as well. So
yeah, it's not.
270 or so degrees C.
Yeah.
So much warmer and about 3,000 meters deep.
Right. I
we
remember
definitely want to keep an eye on this mini Zeus camera then.
Yes, that indicates. That's exactly what we saw last cruise, and we had to switch the fibers.
Copy that.
Yep. At the black smoker.
Look at that mound in the background. It looks like
it's just covered. Yep, covered with them.
Movement right there.
Yeah, it was nice.
That's some fine control.
So for scale, folks, those two red dots that you see are 10 centimeters apart.
All yours.
So that's about 4 inches, and so we can tell how big those snails are. It looks like they are maybe 4 centimeters, 5 centimeters in diameter.
Or the shrimp that you wanted to look at?
No. Pilot, we wanted to look at the snails.
Ooh, looks like there was one data
the round
point
things,
up at
those round little sort
of bottom.
tannish ones, the ones at the bottom, or the ones that are on the pinnacle.
It's good to know.
Also.
See the ones down here because they won't be as much
in the shimmering, right?
48.
Good idea.
Yeah, it was one data point.
Thank you.
High at 48. I'd say there was.
Is that an outlier?
Okay, go ahead and zoom in video.
Yeah, so they're
say a solid 30.
probably two different kinds, the hairy ones and the smooth ones.
And some crabs crawling across the tops of them.
What is that?
If you could tilt to
Active
the left,
volcanism.
Or just, rotate to the left slowly, we can get the vent in
active
there
hydrothermal
Take
activity.
that auto
I've got a bit more
There we go. Hold on, Tara, I'll get it back in.
No worries.
Can we get a real close zoom? Is that max zoom?
We can get a little closer. I have to be very careful not to get too close.
Understood.
But go ahead, Tara.
Don't want to melt the
Okay.
components of the ROV.
Okay, good.
Oh, beautiful. Really beautiful video. You folks at home, this is a real treat.
Yeah.
Giving you really high definition video on these animals.
Yep. The darker one is a species called Ephemeri nautilii, and the lighter one is Alvinoconcha hessleri.
Probably would you be comfortable if I did a little swap here?
Let's wait.
Tim wants to know if these mounts appear to be made out of sulfide.
See your direction of motion.
Yeah, I was avoiding that question because it's really hard to tell, Tim.
Yeah, well, sometimes we ignore those questions.
I would
Just
guess.
tell you guys on sure.
I would guess that these are metal sulfides. What exactly they are, I don't know.
There's a little crab there.
There is a little crab hiding behind one of the snails.
Now.
We did attempt to collect some of these snails with the Shinkai 6500 submersibles.
There's a vent over there on the left. You can try
and of course, you have to use the manipulator arm to grab them. And unfortunately, a couple of them, the shells were broken. And there were a lot of crabs on that vent. And suddenly there was a scurrying of crabs toward the ones that were broken. I don't think they had much time to suffer the loss of their shells because
okay, let's zoom out. Is there anything else you want to look at, watch leads?
On this cone, no.
Okay.
Did we get the vents on the top, just the whole top feature? We haven't documented it yet.
Another fish too.
Go ahead.
And there's a fish. Yeah, there's a fish in the background.
Lasers off.
Yes. And they're going to take some video of this first.
Oh, okay. That's quite a big crab.
Crabs
That's a different species from the one that we saw just a minute ago, too.
It's not as hard as I thought it was.
It's much bigger claws.
Not quite hot enough to boil it.
Is there any way we could get a close-up of the vent? I could just zoom on the vent and then... it doesn't have to be stable, just...
where are you looking exactly?
Oh, maybe the top one that... Just so we have a good shot of the fluid coming out.
Go ahead.
So it's very surprising to not be seeing shrimp, although I'm seeing something on the other. Yeah, I'm seeing some little I'm seeing some tentacles on the back side. Yeah, I can see it right now. Look at those little looks like tentacles there. Maybe they're on the back side. Yep, they're. You think coming? Is there one coming up?
Okay, how's that, Tara? Good?
Thank you, Pilot. That'll be really helpful.
Good. All right, I'm going to move out. Scope ahead.
Okay, so that eel could have been a cutthroat eel. Yep.
Sorry, watch that. It was just really stable. Oh, there's another one there.
Yeah.
You want to zoom in, Tara?
Yep.
Hold there. Zoom in more. Let's get an ID. Okay, let's zoom out. So that was 3.
Oh, the last move?
Yeah.
It's 305.
That was to get me on top of you. I think the
the move before that.
Oh,
310.
Yes, 310.
310. Okay. Yeah, definitely looking down slope. Yeah. Come up.
I'm seeing.
So I'm thinking we might be able to just like we could do like these 30-meter hops, and we can kind of push out and then kind of keep the bottom in sight, come down, take a look around, just slowly hop down.
Yeah, that sounds good.
So let's do another. Let's bring us toward waypoint 3, 310.
No, 30 meters. You got it.
Yeah, 30 meters.
I'll keep my delta around, or my altitude around 20 or so.
Yeah, that'd be great.
I got you at 53 degrees, so we're in a happy spot right here.
All right. Very happy. Just keep an eye on my tail end here.
Yeah, copy that.
Bridge, RV nav.
Taylor looks nice and happy.
Okay.
Hey, we're ready for another move. That's 30 meters, 310 degrees, 0.2 knots.
I'm plotting your temperature, so. On the wing.
I think on the.
It was on the box.
Copy that.
The white fish.
I think it was on the box.
There's
when you're going to
another
stow
fish
it,
in the
yeah,
distance
angles
there,
will off.
lighter colored one.
That's right.
And another one down at about 3 o'clock.
Just above that boulder.
Yeah,
they're off in the distance. They're
You
very
want to take a look
similar,
at those?
guys.
Sure.
Zoom in.
Yeah, sure.
Hold there.
Nice.
Oh, is this a rat tail? Let's see what they say. Bruce Mundy, scientists stumped a